Let's go ahead and get started. This is it, the penultimate, there you go, lecture, our second to last lecture. I should just stop this, delete this recording and start over again. But anyway, today is Thursday, December 2nd. We are in our second to last class. We are going to finish up our discussion of the female reproductive system and then set ourselves up for Tuesday when, like I said, we're going to talk about development. Again, you can literally spend a whole semester talking about development. So we are going to look at the very beginning and the very end and just wave our hands magically at the stuff that happens in the middle. You have two more assignments, one more unit review, and one more labster that need to be completed. And then it's just all tests all the time. Thursday, the 9th, you will have your lab and lecture exams, 2 exams, both on the same day, same format as what we've been seeing for the previous one. So again, no surprises, you know exactly what to expect from that. Then you get a nice long weekend and come back on Tuesday for the final exam. Again, the final exam is 100 multiple choice questions. So again, the huge advantage to that is you don't have to pull the information out of the ether. You just have to recognize the correct answer, which is going to be sitting in front of you. So 100 points for that 100 multiple choice questions, covering everything from the beginning of the class till now, or actually technically till Tuesday. And then just like a final on campus, you'll have 2 hours to complete it. And then you get to sleep. No more anatomy and physiology. All right. Questions on any of that? All right, excellent. Let's pick up where we left off. And it was the end of the day and it was getting a little long. And so I want to go over this again briefly. We were working our way through this table. And again, we had started, well, really, we started with oogenesis, which isn't even on here. But we know that oogenesis is indeed related to follicular development. And so we talked about all of the phases of follicular development that occur during the ovarian cycle. And we left off talking about this complicated relationship between the hormones that influence the follicle and then the hormones that are produced by the follicle. And then the fact that the hormones that are produced by the follicle have a very complicated relationship with the anterior pituitary and the hypothalamus, influencing their production and their release. And that's once the hormonal system, once the cycle is established. We didn't even talk about what has to happen to establish the ovarian cycle. As we mentioned, the ovarian cycle typically starts around puberty. And the reason there is this delay until puberty is that the ovaries, while they're not tremendously active in the production of follicles, they are producing and releasing a very small amount of estrogen and a small amount of inhibin. And so those two hormones play a big role in helping to, as we know, inhibit the hypothalamus and inhibit the anterior pituitary so that they're not producing follicle stimulating hormone and they're not producing luteinizing hormone. But then the female's body gets larger, it matures, it gets healthy enough to be able to produce offspring. And one of the primary ways that the female's body shows the hypothalamus that it is mature enough and also healthy enough to be able to not just produce eggs, but then to house that offspring and bring it to development and birth, is the presence of the hormone leptin. And can someone remind me what makes leptin again?
Adipose cells.
Adipose cells, absolutely. It is the adipose cells that are responsible for producing leptin. So when the woman is both old enough and also physically fit enough, healthy enough, by showing that they are not malnourished, by showing that they have sufficient resources to become that baby oven that is going to be necessary for the next 4 weeks, the leptin that is being produced by the adipose stimulates the hypothalamus to start producing and releasing follicle stimulating hormone. You may not have thought of it in these terms, but maybe you have a friend who is one of those endurance athletes who just for a whim on a weekend will just see how long they can run for 48 hours straight, or have one of these entire entire California, relay races or something like that they do. Some of these female extreme athletes, when their body weight gets down to, 1%, 0.5%, what can sometimes happen with these elite athletes?
They'll lose their period.
Yeah, they'll stop menstruating. Absolutely. They'll stop, their cycle will stop because the inhibition has become so strong without that leptin, without that adipose. You're not getting that extra stimulus to maintain a normal cycle. Now, typically when this cycle is first, the first onset of it, it can be very irregular. They can have their first period and then months can go by before they have their next one. The timing for the cycles can change and vary dramatically. And also through this developmental time, they may not necessarily ovulate during each cycle. However, as we talked about, theoretically, again, we don't want to put it into practice, but theoretically, is it possible that a female can ovulate during her first ovarian cycle and get pregnant on her very first ovarian cycle? Yeah, we hope it doesn't happen, but absolutely it can occur. So there's a tremendous amount of irregularity in the system. And again, often they're infertile. They won't ovulate during that time, or will ovulate irregularly. They can ovulate, skip an ovulation, or the ovulation could be offset and actually occur during menstruation. So when you're young, there's much more flexibility, much more variability, which again is one of the things that makes teenage intercourse so scary, because again, of that irregularity of the ovulation, that if they can ovulate during menstruation, there is the potential that they could get pregnant, even having intercourse during menstruation. But typically, although I know from the last classes we talked about, there can be some exceptions, but typically by the time we reach adulthood, we land on a fairly stable cycle. Again, that can vary from person to person, and everybody's cycle is going to be slightly different, but eventually for most people, they establish a fairly regular cycle. As we mentioned, and again, we went through it with the pretty pictures, but let's go through it with the words, make sure we understand. Again, our gonadotropin releasing hormone stimulates the release of follicle stimulating hormone and luteinizing hormone. Our gonadotropin releasing hormone, of course, is released by the hypothalamus. And of course, follicle stimulating hormone and luteinizing hormone, our gonadotropins, are released by the anterior pituitary. And primarily, our follicle stimulating hormone, as its name indicates, stimulates the follicle to grow. As we start producing those granulosa cells, what do the granulosa cells produce?
Androgens?
Well, okay, the thecal cells produce androgens, but what do the granulosa cells convert them into?
Estrogens.
Estrogens, absolutely. So now they're going to start producing estrogens and also inhibin as well. We didn't talk about that last time, but remember, we don't want this process going too fast as well. And as we mentioned, those estrogens have a very complicated relationship with the anterior pituitary. Remember, inhibin, like in males, primarily targets the hypothalamus, but also can target the anterior pituitary as well. But as we mentioned, estrogen has a very complicated relationship with the anterior pituitary. When the levels of it are low, it inhibits the release, but not the production. So it continues to produce more and more of them. They're just not releasing them from the anterior pituitary. This means the levels of these hormones in the blood actually level off. Or remember, as we talked about with follicle stimulating hormone, it can actually cause it to drop. And again, that scarcity of follicle stimulating hormone is what leads to the elimination of most of the follicles. Because remember, as those follicles are trying to grow from primary to secondary follicles and from secondary to ultimately a tertiary follicle, they have to have that follicle stimulating hormone. Without it, they're just going to degenerate and they're going to go away. So like I said, some get lucky enough to get more than others. Some are going to be more sensitive. So less follicle stimulating hormone will have a bigger effect. But for whatever those individual variations, as we talked about, this is what leads to that race, where one of them ultimately is going to win and outcompete the others. And that one grows huge, and that one matures, and that one contains thousands of granulosa cells. And when you have this big, huge ball-shaped glandular structure that is producing massive amounts of estrogen, ultimately estrogen hits this critical point where now, instead of inhibiting the anterior pituitary, it actually stimulates it. So at this high level, it actually stimulates the anterior pituitary, causing the anterior pituitary to release a massive amount of both follicle stimulating hormone and luteinizing hormone. However, whereas was the follicle stimulating hormone that is primarily important for the follicular phase, it is the luteinizing hormone that is vital for basically two events. That massive release of luteinizing hormone causes two things to occur. What are the two things that occur because of this huge spike of luteinizing hormone?
Ovulation.
Ovulation and?
Formation of,
you got it.
what? L starts with L. I forgot.
Yeah, well, the luteinizing hormone makes the corpus luteum, right? It's a luteinizing hormone. Absolutely, absolutely correct. It triggers that ovulation, and the remaining follicle cells are then converted into that corpus luteum. Corpus luteum is a big, huge, massive glandular structure. And the big difference between the corpus luteum and the granulosa cells is granulosa cells just produce estrogens. Whereas, and really this should be changed. Hold on. I don't like that. Nope, I didn't mean to stop sharing, but that's okay. There we go. Classes now. Produces progestins, primarily progesterone, and estrogens, again, primarily estradiol. So again, the big difference with the corpus luteum is now we're producing those progestins. And remember, the progestins are the big warning bell signal telling the female's body that the egg has been released, fertilization can occur any minute, we're about to get pregnant, get ready. However, these estrogens and progestins are going to inhibit both the production and release of follicle stimulating hormone. So remember, estrogen by itself just inhibited the release, but the estrogens and the progestins together, they inhibit both production and release. So just as rapidly as our luteinizing hormone levels went up, our luteinizing hormone levels start to drop. And that's a problem because the luteinizing hormone doesn't just form the corpus luteum, but it also maintains it. And so if corpus luteum levels go down, or corpus luteum gets smaller. And if our corpus luteum gets smaller, it produces less estrogens and progestins. And as it's producing less estrogens and progestins, as those levels drop, then our anterior pituitary is no longer inhibited. And when it's no longer inhibited, it can start producing follicle stimulating hormone and luteinizing hormone again. And the whole process starts over again. Again, no new information here. This is what we did at the end of the last class. But again, this is a lot more complicated than what we saw in the males. So I wanted to make sure we understood this. And again, now we've done it with my drawing it on the screen. Now we've done it with the pretty words. And of course, now we've got some pretty pictures from your textbook that shows all of this process as well. Here we have these great pictures showing again how that follicle stimulating hormone is stimulating the production of estrogens in those granulosa cells, causing our follicle to grow. It ruptures, it releases the estrogens and progestins, and again is inhibiting the hypothalamus and the anterior pituitary. So here we see with the pretty picture. And then lastly, here we see that graph. As we talked about, with little to no major follicles, there's little to no estrogens. And so we get a rise of our follicle stimulating hormone and luteinizing hormone. But as our follicles grow, that estrogen is increasing and our levels stop being released, but still are produced. And as we mentioned, we even see a drop in the follicle stimulating hormone, which leads to the production of just one mature follicle. At this point, we're producing a massive amount of estrogens, and that massive amount of estrogens actually excites the anterior pituitary, and we get this massive release of both luteinizing hormone and follicle stimulating hormone. And again, that big, huge spike of luteinizing hormone does two things. It triggers ovulation and it produces the corpus luteum. Of course, not only does it produce the corpus luteum, but it maintains it. So as these levels start to drop, our corpus luteum starts to degenerate until it shrivels up into that little piece of scar tissue. And what do we call that little piece of scar tissue at the end of the process?
Corpus albicans.
Yeah, corpus albicans, right? The white body, corpus albicans, white body. So it's just a little bit of scar tissue stuck in there at the end, at which point now we can't make any estrogens or progestins anymore. And so lo and behold, our inhibition is released and levels can start coming back up again. And the process continues. And here we see those estrogen and progestin levels that we were talking about. All right. Questions on that? We've now covered 3/4 of this graph. That was very scary when we first looked at it, but now it's totally making sense to us. Or at least 3/4 of it is. Questions on that? All right. Now that we know what's happening with the ovary, let's talk about the rest of the reproductive system, or more specifically, the reproductive tract. The reproductive tract of the female starts with what is known as the uterine or fallopian tubes. You may hear both of those terms. They are connected on the lateral aspects of the uterus. There are these long muscular tube-like structures that have 3 main specializations to them. These 3 main specializations are an enlarged portion right at the proximal end, known as the ampulla, That's a term we'd heard before. Big, large open space is an ampulla. And it is actually in this ampulla where the egg is most likely to be fertilized. Off of that ampulla is a funnel shape structure. This funnel shaped structure that feeds into the ampulla is known as the infundibulum. So over here we have this funnel shaped infundibulum that feeds into the ampulla. And this funnel shaped structure then has these finger-like components that extend off of the infundibulum. And these finger-like extensions are known as the fimbria. So if those are the fingers, the infundibulum would be the palm, and then the ampulla would be, I guess, the wrist then, if we were going to think of it that way. These fimbria do not actually attach to the ovary. Notice they drape over the surface of the ovary. But remember also, as we talked about, when ovulation occurs, the follicle that is released could be located right here, or could be located right here, or could be located right here, or right here, or any spot on that ovary. So the fimbria are not actually connect. They're not tubes that connect. They really are these finger-like structures. And these finger-like structures move in a wave-like pattern. And as they move in this wave-like pattern, they produce currents in the abdominal pelvic fluid. And the goal of those currents is to, when the egg is released, to draw it into that funnel-shaped infundibulum, and from there into the ampulla, where it will wait for the sperm for fertilization. Now that is the goal, but is it always going to get into that infundibulum?
No.
No. So there are going to be plenty of times where it's going to be left floating around out here in the not interstitial fluid, but in the peritoneal fluid, within the peritoneal cavity. So again, is that likely to be fertilized by a sperm if that is the case? No. Can it be? Yeah. Theoretically, a sperm could swim up the uterus, up the uterine tube, out the uterine tube into the abdominal pelvic cavity. And it's possible you could get enough sperm to that egg to fertilize it. Maybe. It's not very likely, but theoretically it could happen. Theoretically, though, if it happens, is that fertilized egg going to be able to find a location where it can find purchase, and as a result of there start to develop and grow?
No.
No. Except in very rare cases, it might. It might find a location where it is actually able to implant and start to grow. But if it were start to grow there, would you be able to bring that to term as a viable baby? No, absolutely not. Right? There's no way that that would be able to grow to term. The baby would definitely not survive that. And most likely, mom wouldn't survive that as well. What do we call that condition when that occurs? There we go. Amy's got it. Nectopic pregnancy. Very rarely will it happen outside of the reproductive tract. Occasionally, slightly more commonly, it'll actually occur in the uterine tube. The egg will make it into the uterine tube. It'll get fertilized in the uterine tube, but it accidentally implants in the uterine tube. And again, while it may start growing there, is it possible for that baby to be brought to term and for the baby and for the mom to survive that experience? No, absolutely not. And but don't worry, if the Supreme Court has anything to say about it, we'll make sure all those women die for the mistakes of not fertilizing their egg in the wrong place, in the right place. So they'll take care of that very, very shortly. Now, the one bright spot is you live in California. Hopefully you all live in California. I guess with this being online, technically not everybody here is in California, but hopefully you're in California and your reproductive rights as a female are safe. Just don't travel the country anymore. All right. So fucking stupid. All right, questions on that?
So if it gets fertilized outside, like in the peritoneal cavity, does it still develop placenta?
No. Like I said, in most cases, it would not be able to implant. So in many cases, it would just basically eventually reach a point of not being viable anymore and would degenerate and just be absorbed by the surrounding tissues. In rare instances, it can find a location where it just happens to connect to a piece of connective tissue where there happens to be a good enough blood supply where it's able to start to grow for a period of time. But to get to the point where you would produce a placenta is about 9 to 10 weeks. And in all likelihood, wherever it found to find purchase, I would not be able to grow and implant and expand and enlarge enough to get to a point where it would be able to form a placenta. You would probably have much more complications where it would reach a stage where it's not capable of maintaining itself anymore, or where it's causing complications and damage to the female to a point where you'd be made aware of it, become aware of it, and it would have to be removed before it would have a chance to. And again, even if you left it alone, there is almost a 0% chance that it would be able to grow to a stage that it formed a placenta. And even if it formed a placenta, there is a 0% chance that it would be able to be carried to term and be viable. Yep. Believe it or not, there are actual reasons why a fertilized egg might need to be terminated. All right. Questions on that? All right, so that is our anatomy of the uterine tube. Oh, I guess I didn't mention the narrow region where it connects to the uterus is known as the isthmus. So I guess I forgot to mention that. And again, it is directly connect to the uterus, but as we mentioned, it is not directly connect to the ovaries. Again, this is an open duct system. Here we get a little bit of a better view of the components as we talked about for that uterine tube. Again, we have the enlarged ampulla where fertilization will take place, the funnel-shaped infundibulum, and the finger-like projections, the fimbria. Now, if you remember, in the epididymis of the male, we found a epithelial tissue that we hadn't seen before. We saw a non-motile ciliated pseudostratified columnar epithelial tissue, right, in the epididymis. We had a non-motile ciliated pseudostratified columnar epithelial tissue. As we mentioned, it was a unique tissue, and that was the only little place that we find it. Here in the uterine tube, we find a unique tissue we haven't seen anywhere else in the body either. It is a ciliated simple columnar epithelial tissue. Normally, when we think of a simple columnar epithelial tissue, we think of them having microvilli. But here in this case, we actually have cilia. And those cilia help to move the egg through the uterine tube. Obviously, the egg doesn't have a flagellum the same way that the sperm does, and so requires assistance in getting to its location. So obviously, that is going to increase surface area, allowing for the maintenance and support of that egg, but also for the movement of the egg as well. So that's really its goal. The function, and really it should be functions plural. Obviously, the first one is to catch the ovulated oocyte. House it for fertilization. And then move it to the uterus for implantation. Now, these cilia that are going to move the egg towards the uterus take around 5 days. 5 to 7 days to reach the uterine tube. Let's actually say it that way. To go through the uterine tube to get to the uterus. However, when that egg is ovulated, it is only viable for about 24 hours. So notice the only way that that egg ever gets to see the uterus is if it is fertilized. If it is not fertilized, it is going to degenerate and be absorbed by the tissues well before it ever reaches the uterus. I make a point of emphasizing this because I know I've had students in the past who thought that during menstruation, part of menstruation was getting rid of that unused egg. That is not the case. The oocyte does not reach the uterus unless it is fertilized. If it's not fertilized, it will be broken down and absorbed in the uterine tube well before it reaches the uterus. All right. Questions on that?
So it's only fertilized in the uterine tube?
Yep. Yep. So like I said, 3 to 4, it's really more closer to 5 to 7 that it takes to move towards the uterus. Let's talk about the uterus. The uterus is a hollow organ located between the bladder and the rectum. As I mentioned, in a female that has never been pregnant before, it's about the size and shape of a pear. And we see that nicely here. Well, at least an upside down pear. Notice the narrow inferior region is what is known as the cervix. This is a well-muscled region with a narrow opening that passes through it, known as the cervical canal. Notice also the head of the cervix actually extends out into the vaginal canal. If you were to reach into the vaginal canal, you can actually palpate the cervix. In fact, that is what a doctor does when he is checking to see how far a female is dilated. Basically, he is feeling for two components of the head of the cervix. They're looking to see how wide that cervical canal has dilated, right? Because again, to pass that basketball, it's got to go to the massive, massive size. I don't know if I have a ruler. I don't have a ruler. 10 centimeters, right about there. So again, huge and massive, easy to pass a basketball through that, but also how pliable, how much effaced it is. So they measure the pliability of it, they measure the size of it, and that is the head of the cervix that is sticking out there. The majority of it is, of course, the body, which is a large central region. And then the superior part is a big enlargement known as the fundus. Nice big round fundus, kind of like the stomach has a fundus. Nice big enlarged round part. That's what fundus means. What's interesting about the fundus is that as the baby is growing inside of the female, obviously the uterus has to grow as well. And as the uterus is expanding in the abdominal pelvic cavity, again, this is something that can be felt. You can actually, through the stomach, feel the fundus, the fundus, the top of the uterus. And what they will actually do is they will measure from the pubic symphysis to the top of the fundus. And that distance from the pubic symphysis to the top of the fundus is probably the strongest indicator to how far along a female is in their pregnancy. It is how they may take the most, other than obviously an ultrasound where we can see the internal development. That is obviously the end all be all because we can actually see the baby itself. But that measurement from the pubic symphysis to a fundus is just about as accurate, almost as accurate as actually looking at the developing cells themselves. So it's a pretty cool characteristic of the uterus as it grows, that it has such a consistency to it. Again, its job is to receive that fertilized egg. That fertilized egg will then implant itself into the wall of the uterine tube, and then it nourishes it and maintains it as it grows and becomes a baby. And then ultimately, the uterus is then responsible for expulsing the baby out into the real world, where it can get a job and make you grandkids. All right. Questions on that? Like most of the organs that we have seen, it has three main layers. Notice, kind of like the digestive system, and actually let's do this on the whiteboard. Let's start easy. What, of course, is the middle layer going to be?
Myometrium.
Absolutely, is indeed called the myometrium. Excellent. And what type of tissue is it?
Smooth muscle.
Smooth muscle. Right. Again, can you voluntarily contract your uterus on demand? Bend it to the left, bend it to the right, twist it left and right. No, of course not. It's smooth muscle. We have involuntary control of it, but it has a big, thick layer of smooth muscle at the center of it. Then if we think of the lumen is connected to the outside world. So what would we have? What kind of membrane would we have on the inner surface? If the lumen's open to the outside world, what it lines cavities that are open to the outside world?
Non-keratinized, stratified, squamous, epithelial.
Well, okay, you're thinking in terms of what kind of epithelial tissue and connective tissues form the membrane, but yeah, I like where you, so you're going with the right idea, but what actually type of membrane do you think it would be?
Oh, mucous membrane.
Yeah, it's going to be a mucous membrane, absolutely. So we're going to have a mucous membrane on the inner surface. And what are we going to call this mucous membrane on the inner surface? There we go, an endometrium. Excellent. We'll talk about what epithelial tissue and connective tissues are there in a second. But then we also have an outer layer. Here, let's go ahead and draw this. So there's our mucous membrane on this side. And then we're also going to have a membrane on the outer surface. This membrane, again, this is an intraperitoneal organ. So that should make it easy. What type of membrane lines the outer surface of our uterus? Well, if it's not mucus, peritoneum, which is a serous membrane, or a serosa. And you're absolutely right. It will be specifically the visceral peritoneum. Excellent. Right? Because it's an intraperitoneal organ in the abdominal pelvic cavity. And of course, like all serous membranes, we know that it's got an areolar connective tissue. And the epithelial tissue is simple squamous, which we would then call a mesothelium based on its location. And of course, it's got a thin line of transudate, that's serous fluid, on its outer surface. Excellent. Makes it easy up here because we know for our mucous membranes, it's always also going to be in a areolar connective tissue. And again, this one, based on its location, we call a lamina propria. I like where you're thinking with the non-keratinized stratified squamous epithelial tissue, because like we said, openings that are open to the outside world need to be protected. However, in this case, remember, it's a little bit further away from the outside world, and we want our egg to be able to penetrate into it and be able to implant itself within there. So would having many, many thick layers of cells be conducive to getting that egg implanted inside? No. So instead, the outer surface is just lined with a simple columnar epithelial tissue. So we just have a simple columnar epithelial tissue sitting on top of an areolar connective tissue. However, this endometrium is actually further divided into two distinct, well, let's not use blue. Two distinct sublayers. The stratum functionalis. I'm gonna have to make this smaller. Or what is also known as the functional layer. And the stratum basalis. Or the basal layer. There are several differences between these two, but probably the biggest is that the stratum functionalis growth and development is strongly related to hormone levels. And so it changes much more dynamically. Whereas the stratum basal is essentially not sensitive to hormone changes. And we'll see why that's important in just a minute. Let's go back here. As I mentioned, we have our inner layer, that endometrium. This is what allows for implantation. And this is also going to be the layer that is going to slough off and be released during menstruation if pregnancy does not occur. We have our myometrium, which is that smooth muscle layer. And again, that smooth muscle, remember, as we talked about, is going to produce peristaltic contractions during normal function of the female to move substances out, keep it out of the abdominal pelvic cavity. But remember, we talked about how orgasm and then also the prostaglandins in the semen can cause a reverse peristalsis to try to draw the sperm up towards the uterine tubes. And then the outer layer, our visceral peritoneum, our serous membrane, of course, because anatomists hate us, we'll give it a third name as well, a perimetrium. So if we cheat and go back here, we can add a third name. But again, perimetrium is just a fancy way of saying serous membrane, fancy way of saying visceral peritoneum of the uterus. So perimetrium, myometrium, endometrium. As I mentioned, the endometrium has two sublayers to it, two specific layers, the stratum functionalis, or the functional layer. This is the part that changes in response to our ovarian hormones. This is the part that is shed during menstruation. Whereas, as I mentioned, that stratum basalis is insensitive to hormone changes. As its name states, it's the base, it's the beginning part. And once the functional layer is shed, it is from this base, from this foundation, that we grow a new stratum functionalis. This also houses most of the uterine glands, which also grow and expand dramatically during the uterine cycle, which is something we have not talked about yet. Last little bit of anatomy we have to look at here. Here we see this histologically, and I have some really good histology slides that I will share with you a little bit later as well. But again, here we can see really nicely the distinct smooth muscle layer, all of this smooth muscle, irregular banding of the myometrium. Then basically within here, we can see this darker, more cell dense region where there's some beginning of some large glands located. That is the stratum basalis, that basal layer. And then we can see up here that growth of the big, huge functional layer that is growing and expanding in response to our uterine hormones, ovarian hormones, sorry. We've got another pretty picture that shows this here. Again, notice it's a little bit subtle, where again, the denser, darker portion that is close to the smooth muscle is the stratum basal, and the stratum functionalis is the area above that. And the reason they did it more subtle here, and I'm going to go ahead and erase it now that I've shown it, is because the point of this is to show that not only is there a very elaborateness to the tissues, but there's also a very elaborate blood supply. Now, as I'm sure you remember from cardiovascular system, most of the organs in the pelvic cavity, the pubic organs, are fed by the internal iliac artery. Off of that comes the uterine artery. And that uterine artery feeds into a large arcuate artery. Now, I'm not going to hold you responsible for that pathway. But what I do want you to see, and what is important, is that in the stratum basalis, there are two major types of blood vessels. There are let's do it this way. There are these short, stumpy branched blood vessels known as the straight arteries. Their job is to primarily maintain the basal layer. But notice also there is a much more elaborate, curving, rotating, coiled-like blood vessel that then branches very elaborately and is very large. And this is what is known as the spiral or the coiled artery. As you can see, this is the one that is going to have the expansive growth to be able to feed the functional layer. But here's the problem with our spiral artery. The spiral artery is very sensitive to hormone levels. And in fact, primarily estrogen, but also progestins as well. And when estrogen levels drop, the spiral artery kinks. And what happens when you kink a hose?
The flow gets stuck.
Yeah, the water doesn't flow through it anymore. So guess what happens when you kink this spiraled artery?
The blood supply is cut off.
Yeah, blood no longer gets up here to the functional layer. And so when the hormone levels drop and this blood vessel kinks, can we provide the oxygen and nutrients necessary to be able to maintain the functional layer of our uterus? These are the easy questions, folks. No. And so this is when it degenerates, it sloughs off, and it is released via menstruation. So it is these two main types of blood vessels, the straight, which aren't sensitive to hormones, so they're always feeding the basal layer, and that never changes. But they're also not very dynamic. The spiral is very dynamic, allowing for big, elaborate growth, but it's also sensitive to those hormone levels. So if estrogen levels drop, it kinks, and we can't maintain that functional layer anymore. And really, in a nutshell, that's kind of what the uterine cycle is all about. Right? Those functional layers dies, the straight arteries are there to help to start the regrowth, and the whole process begins again. Of course, we have to be more specific than that. So of course, we will talk about the uterine cycle, and we'll do that next. But even though it's a little bit early, this is a good natural stopping point. Let's go ahead and take our first break here. When we come back from our first break, we will do our uterine cycle. All right, 15 minutes. Looks like it's 12:53. So let's come back. Oh, what'll that be? 107. And we will restart at that point. This here. And then I'll start the recording at that point as well. All right, questions on any of that? All righty, I will see you guys in 15 minutes. Let's go ahead and get started. Our goal now is to finish up our last main related cycle, and that is the uterine cycle. These are the changes that take place primarily in the endometrium. And again, it is occurring in synchrony with the ovarian cycle because our uterine cycle is controlled by the ovarian hormones. So as the hormones in the ovaries that the ovaries are producing expand and change, those cause the expansion and the change of the uterine cycle. Now, remember, as we mentioned, there we go. At the very beginning of our 28-day cycle, all of our hormone levels, not just follicle stimulating hormone and luteinizing hormone, but remember, primarily estrogen and progestin levels are low. And when those levels are low, right, as we talked about, that spiral artery is going to kink. We are not going to be able to maintain the functional layer of the endometrium anymore. And that functional layer is going to basically break off, slough off, and be expulsed. Peristaltic contractions of the uterus and vaginal canal lead to the expulsion of the menses. The menses is primarily the functional layer. And I know it may not seem like it, but only about 35 to 50 milliliters of blood is lost during this process. Now, again, I'm not talking about 35 to 50 milliliters of stuff. Obviously, the lining of the uterine wall is a large mass. And as we mentioned, remember also after a female has been pregnant, typically the uterus doesn't get back down to the size it was originally. So it is not uncommon for the size of the menses to increase after childbirth because of that increased amount of tissue. But it isn't a major source of blood loss, because as we mentioned, with the kinking of those blood vessels, not a lot of additional blood is lost, but the blood that is already engorging that tissue, along as the tissue itself, is expulsed. And this process typically takes 1 to 5 days. Again, during this time, we are not seeing major growth inside the ovaries. We're just starting to get that development from the primordial into the primary stage. And so, as we mentioned at this point, estrogen levels are low. But then, as we know, those primary follicles become secondary follicles and become a mature tertiary follicle. And as our follicles continue to grow, the amount of estrogen increases. And as those estrogen levels increase, we enter what is known as the proliferative stage, proliferating, making more of, or obviously what is known as the pre-ovulatory stage, because it is occurring before ovulation. Again, remember, as I mentioned, this is the more variable. I thought I had that written here. We've got here as the D6 to D14, but as I mentioned, this is the more variable stage in this process. And again, it is driven by the estrogen. That functional layer is regrowing from the basal layer. As it is regrowing, those spiral arteries, even though we don't see that histologically here, those spiral arteries are going to start to regrow and re-expand into the tissue. And the uterine glands. Remember, we said the base of the uterine glands are located here in the basal layer, but the glands are going to start to grow back. And that's all of these structures we see here. Notice, as we mentioned before, the epithelial tissue on the outer surface is simple columnar. But we also see a lot of simple columnar and simple cuboidal cells located within the functional layer. And these are all of the glands that are regrowing. These glands produce a very glycogen-rich mucus secretion that, if that egg comes to the uterus, will be maintained and we provide the nourishment and the energy necessary to continue to grow and divide and get ready for implantation. This continues for, like we said, about 10 days or so. And again, it varies from person to person until that magical event, ovulation, takes place. Again, in a typical 28-day cycle, that occurs at day 14. At day 14, and again, here's what I want you to do real fast before we switch screens. Notice here is our myometrium. Here is the top of the tissue. So the entire stratum basal, the entire endometrium and some of the myometrium is in our field of view right now. And notice, even with a naked eye, we can pretty much make out the individual cells, for instance, in the tubule. We can see the nuclei on these simple columnar cells. They're not perfect. Some places it's better than other. Here, we can see the cells pretty well. But notice the relative size of these structures. I make a point of emphasizing this because when we hit the third phase, the secretory phase or post-ovulatory phase, this is when we get massive, massive growth of the functional layer. Notice all we see now in the field of view is the functional layer. Here's the top of part of it, although it looks like it could be expanding more out that way as well. We don't even see any of the basal layer, let alone any of the myometrium. And maybe the cells are a little bit smaller, so this might be a little bit of a lower magnification. Remember, the lower magnification we can get more of the field of view in here. So we are seeing massive, massive growth in the functional layer. And this functional growth is caused by that progesterone. Those progestins that are being released, that warning bell, as we mentioned, is the warning bell to tell the female body, get ready for pregnancy. And the primary thing that needs to get ready for pregnancy, or at least it needs to get ready for pregnancy first, is the uterus. Massive growth in the size of the functional layer. And look, also very distinctly, you can see a lot of the spiraling of the glands. This big, distinct spiraling is very consistent within this functional layer when we're producing these big, huge, massive glands that are producing that big glycogen-rich uterine milk. That's basically what they call it, in anticipation of that fertilized egg coming down into the uterus. And let's do some math. As we talked about, it could be a full day, 24 hours before that egg gets implanted. Egg gets fertilized. Once it gets fertilized, it can take anywhere from 3 to 5 days to get to the uterus. Once it gets to the uterus, it can take an additional 3 to 5 days before it implants into the wall. So while this is getting ready, it can take as long as 10 days before the egg, actually the fertilized egg, implants into the wall of the uterus. So it needs to get ready, and it needs to stay ready for a good week and a half in anticipation of that egg finally getting there. But day 10 comes, still no egg. Day 11, 12, 13, still no egg. And notice, if that egg wasn't fertilized, as we know, our corpus luteum is going to start to go down. And as our corpus luteum starts to go down, we are going to see a decrease in the progesterone levels. And as we talked about, that will lead to the kinking of our spiral artery. Oh, that have it there. So low levels of estrogen and progesterone lead to the kinking of the spiral artery. And the process continues. And notice, this is our last illustration on this chart. Notice again, at first, estrogen and progestin levels are low, and the functional layer is sloughed off. Then, as estrogen levels start to increase, we start to get the regrowth of the functional layer. Once ovulation occurs and our corpus luteum forms, and now it starts releasing progesterone, progesterone not only leads to a massive growth, but notice also a massive vascularization, the spread of those spiral arteries, and the massive glandularization. Because that egg, when fertilized, when implanted, is going to need a ton of oxygen, a ton of nutrients from that blood supply. It's going to need glucose and all sorts of other chemicals from that uterine milk. It's going to need to be maintained. So all these structures are vital for the success of that egg. Of course, if the egg doesn't get fertilized, then our luteinizing levels go down, our corpus luteum degenerates, estrogen and progestin levels drop significantly, and our blood vessels kink, and the uterine wall, the functional layer of the uterine wall, cannot be maintained any longer. It sloughs off, and the whole process begins again. As we mentioned, this process establishes itself at puberty, and then once it's established, goes till menopause. A continuous cycle. And in between puberty and menopause, only one thing stops this cycle. What is the one thing that stops this cycle?
Pregnancy.
Exactly. Remember, as we mentioned, when that egg gets fertilized, it produces a special hormone called human chorionic gonadotropin. And what human chorionic gonadotropin does is it maintains the corpus luteum. Again, luteinizing hormone levels drop, and normally that causes the corpus luteum to go away. But now our human chorionic gonadotropin maintains the corpus luteum. So it stays big and it keeps making high levels of estrogen and progestin. And it continues to do so until about week 9. And week 9 is when the placenta forms. Once the placenta forms, the placenta is able to produce the hormones that are necessary. The placenta produces the estrogens and the progestins that maintain the uterine wall and help the baby to continue to grow. And like we said, this maintaining that high level of progestin is basically the way the baby, the uterine wall and the developing embryo are maintained. And as we talk about, as you're maintaining the uterine wall and as you're maintaining that fertilized egg, we don't want to be developing more follicles. We don't want to be ovulating more follicles. We don't want to get pregnant a second time if we're already developing the first baby. So as we mentioned, the very first birth control pills were basically progestin supplements. Trick the female body into thinking it was pregnant so it wouldn't ovulate more eggs. Now, it's gotten a little bit more sophisticated. There are still progestin pills that women take, typically not as birth control because there are other effects that those progestins can have on the female. So there's more sophisticated ones, but there are still some. However, one of the things that will happen is in certain instances, you may have a couple who are successful at fertilizing the egg, starting the embryo, but the embryo consistently doesn't either implant, or when it implants, it's not able to produce enough, receive enough progestins to be able to be maintained. And so if you're able to fertilize the egg, but not necessarily maintain the pregnancy, then often once that egg is fertilized, you'll start taking progesterone pills to maintain that elevated level of progestins to maintain the pregnancy. So they can get pregnant, they just can't maintain it. And so progestin is very frequently used in those instances to help those people to maintain their pregnancies until the placenta has a chance to form. All righty. So again, a little more complicated than what we were dealing with the boys, but that is the female hormone and biological cycles. Questions on that? Again, all of these make good essay questions on their own independently, but also make sure you understand how these things relate to each other as well. All right. Any other questions on this? And again, oh, go ahead.
Yeah, I've just always heard that you're supposed to like eat more iron when you're on your period. Is that not true?
Well, again, you are losing blood. You are losing both a little bit of blood from the blood vessels and also the blood that is engorged in the tissue. And as we know, Iron is an important component of making more red blood cells. So typically when a female is in menses, there is an increased likelihood of her becoming more anemic, especially since a lot of people don't necessarily get the iron that they need. So if your iron levels are already low and then you enter menses, then that can potentially cause difficulties. So it isn't something that you necessarily have to do, but is it potentially beneficial? Absolutely, sure. Yeah. Not necessarily required, but and again, don't eat a whole half of a cow, but yeah, everything in moderation. But yeah, there isn't certainly any harm, and there's definitely some potential benefits to that. Absolutely. Great question. Any others?
Professor, what can cause the menopause to start early? Like I know someone that started, I think at 27, age 27.
Wow. Okay, so here are the things that I will say about that. As we know, these are, and as we're seeing here, obviously these things are all related to hormones, or a lot of this is related to hormones. And as we talked about before, studying hormones and the effect of hormones that they have on the body is something that is very challenging to be able to do. As we mentioned, it's only about 20 years ago that we realized that the hypothalamus controls the anterior pituitary gland. So there's still a tremendous amount we're learning about the interactions between hormones and how they influence and affect things. The second thing is there isn't just one cause. And again, part of the thing that's happening as our understanding of this is growing is we're learning also that menopause isn't just a line where you cross the finish line and then suddenly your body undergoes this changes. These changes are things that actually can begin years before menopause sets up. In fact, there's more and more research going into the study of this premenopausal phase prior to the onset of menopause and the effects that has on women. And we're understanding that those effects can occur earlier and earlier. All of that being said, 27 seems excessively early for something like that. There are numerous things that could potentially cause it. It could be a problem with the pituitary gland and the gonadotropins that are being produced. It could be increased androgen levels that are being produced by the adrenal gland. I mean, there could be, as we talked about, even things as changes in weight. As we talked about, if someone is an ultra marathon runner, and again, I'm not saying that this is the case with your friend, but people who are anorexic or bulimic, again, because of the loss of leptin that is produced because of the loss of adipose, those are things that can disrupt the cycle as well. So there isn't just one thing that could cause it. I assume if this is something that is happening to her and bothering her, then the best thing to do is for her to see a physician about that. And that's one of the things that in general, usually the thing that I tell students is, because not surprisingly, being an anatomy and physiology instructor, students are always all the time asking me about their conditions. And the one thing that I usually say is, if you're concerned enough about it to be asking me, then the best thing to do is to speak with your physician about it, who has much more history with you and understands your situation and things along those lines. That does seem outside of the norm to have it begin that early. The problem is that there are a lot of things that could be causing it. It could be a temporary thing, like I said, because of the physical changes to their body, or endocrine related, or any number of things could be causing something like that. So like I said, if it bothers her, then the best thing to do is to see something about it.
Yeah, she found out about that after she got she got married, and then she had complications to get pregnant, and the doctor said, I think she only had a few cells left, a few eggs left. So
That's
she
true,
had
yes.
to took those progestins, but she got pregnant and she has 2 boys now.
That's
But
awesome.
I don't know what's how can you have few eggs?
Okay. So remember, as we talked about, so there is an attrition from the onset of puberty, from the onset of birth. Remember, a female has all the eggs she's ever going to have. And while they are locked in those primordial follicles and protected, remember, as we mentioned, there is a huge attrition till we get to puberty. So it goes from 2 million to 4 to 200,000. Then remember also we talked about how every month at the beginning of the month, dozens of primordial follicles will start to mature. And as we mentioned, it can take over a year for them to get to the primary stage. However, it is possible, because of her hormone levels or things along those lines, that if more of her primary follicles are maturing at the same time, then again, remember, as we talked about, only one's going to win and the rest are going to degenerate and go away. So if each month you're basically losing more and more of those eggs from your ovaries, then that would also, again, if you remember, the estrogens that lead to the cycle come from the follicles. And if you don't have a lot of follicles, then you're not going to get a lot of hormones. And so if she's been running through her supply more fastly for whatever reason, that could be that would cause an early onset of menopause as well. Because if you don't have enough follicles left, you're not going to make enough estrogens to maintain a cycle.
Oh, so that's why she had to take progesterone, so the egg doesn't get released, and she had only few left, and she had to take progesterone. So whenever she wants to get pregnant, she will stop taking those, so
Awesome.
the egg can be released.
And are the twins identical, or are they fraternal?
No, they're not twins. She had them like 1 year apart.
Oh, okay, got it. Okay, I misunderstood. Then yeah, so yes, then by taking the progestins in between, she is retarding the maturation of the remaining eggs that she has.
Okay.
So that she's not losing more as she's waiting until she decides to have pregnant. Well, I'm glad that worked for her twice. That's awesome. That's even better.
Yeah.
Yep.
Okay, thank you.
Excellent. Great questions. Any others? This is always a fun section. This is always something that obviously many people are very intimately related to and relate to and understand and have friends or even personal experience with these kind of things. It is a very fun field. And like I said, these developmental classes, if you ever have a chance to take one, I took one in college, and they're a tremendous amount of fun. Really, really interesting and a tremendous amount of fun. The human body is awesome. All right. And when the human body makes another human body, it's even cooler. All right. Again, we've talked a lot about the hormones and everything and where they come and what they do. Your book's got some nice tables, so make sure you understand all of these hormonal interactions. What we need to talk about now is the rest of the female. How are we on time? We're doing good. The rest of the female ductwork system. We went from, again, it's an open system, but we still went from the uterine tube to the uterus, and now to the vagina. Remember, as we mentioned, the head of the cervix extends out into the vaginal canal, and the vaginal canal is basically a muscular tube that extends from the cervix to the exterior of the body. Again, it is behind the bladder, in front of the rectum, and I think I have, do I not have a picture? I don't have a good picture. Here, let's do it this way. There we go. Here's one of the models that's in the classroom. So again, notice, as we've talked about now, both for the uterus and for the vaginal canal, they are between the bladder and the rectum. So back here, obviously, is the rectum. Here is the bladder and the urethra. And then we actually see the entire ductwork system of the female here. We have the fimbria, finger-like extensions of the uterine tube that feed into the funnel shape infundibulum. We then have the enlarged ampulla of the uterine tube, which is feeding into the isthmus, where it enters into the uterus. Here we see the uterus kind of folding in over on itself. So the fundus would be up here. And here we see the cervix that, as we mentioned, extends out into the vaginal canal. And here we have the vaginal canal. Now, as I mentioned, the vaginal canal has its external os, the orifice that is open to the outside world. And remember, at the proximal end, we have that head of the cervix that sticks out. I wanted to grab this picture because back in ancient times, and by that I mean, of course, the 60s and 70s, there was a form of birth control, which basically you would go to your gynecologist, and your gynecologist would measure the shape and the size of the head of the cervix. If you notice, because the head of the cervix extends out into the vaginal canal, there are these two basically folds. We see the anterior and the posterior examples of these folds around the head of the cervix that are known as the fornix. And so what would happen is your gynecologist would measure this for you, and they would produce a medical device, which was basically a metal ring. And this metal ring had a rubber sheet over the top of it. You would then take that metal ring, right? When you have that nice dinner and you're feeling amorous, you would then take this metal ring, insert it into the vaginal canal, and place the ring around the head of the cervix. It would then have that rubber sheet that would extend over the top of the cervix, blocking the opening of the cervical canal. And if you were particularly worried, you could put a little spermicide in here as well. So if any of those pesky sperm accidentally wiggled their way into this structure, then that would kill any sperm that were able to wiggle their way in. Of course, what was this medical device called? Come on, someone's got to know. What'd they call this device? Well, I'll give you a hint. It's got a bell-shaped curved structure.
Oh, diaphragm?
Yeah, it was a diaphragm, absolutely. A diaphragm, when used properly, especially with spermicide, has almost 100% success at blocking pregnancy. Right. So then why aren't they so common today? Well, there's three reasons. The first is, notice the key word I said before, when used properly, right? To use it properly, you have to properly insert it, and you have to make sure it snugly fits over the head of the cervix. If you don't get it fully snugly fit onto the head of the cervix, then it can come loose. And if it comes loose, it's not providing that protection. And again, inserting it when you're feeling amorous is not necessarily the most romantic thing. Hold on, honey, while I shove this thing inside myself before we have sex isn't the most amorous thing. Also, many women were often embarrassed to go to their gynecologist about it. So they would just borrow the diaphragm of their friend. You know, the roommate has a diaphragm. If I'm getting lucky tonight, I'll use it. If she's getting lucky tonight, she'll use it, right? Are these things interchangeable that way?
No.
No, these are medical devices that are supposed to be fit specifically for you. If it's not your diaphragm, it's probably not going to fit snugly, and it's not going to fit correctly. And so they're, won't provide the same protection that way. And again, because like I said, this started in the 60s and 70s, what made it even worse is that this obviously provides no protection for sexually transmitted diseases, right? Basically 100% effective when used properly for pregnancy, but it does absolutely nothing for sexually transmitted diseases. So the ease of condoms, the fact that you don't have to go to a doctor to buy condoms, and the fact that condoms can mostly protect from pregnancy and also are very effective at protecting from sexually transmitted diseases. Not surprisingly, diaphragms have gone out of fashion as a result of that. All right. So we have that fornix around the head of the cervix. We then have the external os, the opening to the outer surface. Notice it is inferior to the abdominopelvic cavity. So it is not a retroperitoneal or intraperitoneal organ. It's really inferior to the peritoneal cavity. So not surprisingly, on the outer surface, it has an adventatia, which basically just anchors it in place. Notice also from this illustration, it has a middle layer, a muscle layer. And what type of muscle would make up the muscle layer of the vaginal canal?
Skeletal.
Skeletal? Can you contract your vagina on command? Let me answer that question for you, because I know some of you are going to think you can. You cannot. Now, I know anyone who's ever gripped their loved one may think otherwise, but what you're actually doing when you're increasing the tension that way is you are actually contracting the levator ani muscle. The levator ani muscle, remember, surrounds the vaginal canal. So when you are increasing that pressure by elevating the levator ani muscle, you're actually using the levator ani muscle. The muscle of the vaginal canal is actually smooth muscle. The contractions of it that occur during menses, the contractions of it that occur via orgasm, the contractions of it that occur during birth, all of those are peristaltic contractions. All of those are involuntarily controlled. You cannot actually voluntarily contract the vaginal canal. Again, you can tighten the opening by affecting the levator ani muscle, but it is actually smooth muscle. This is open to the outside world. So of course it is lined with a mucous membrane. And what type of epithelial tissue will line this mucous membrane? Now you can say it.
Simple pulmonary.
Non-keratinized.
Non-keratinized stratified squamous, right? Remember, here in the uterus, we want to make it easy for the egg to be able to implant into the wall. However, the vaginal canal is a copulatory organ. Its job is to receive the penis, right? In fact, that's where the name vagina comes from. It basically means sheath, like for a sheath for the sword, right? So it is a copulatory organ, and copulation, when done properly, should be vigorous, right? As such, we want to make sure that, again, this is an area that could see a lot of friction. We need protection. And so having those many layers of that non-keratinized stratified squamous epithelial tissue will be present here. Notice something else, the way I've drawn it, because I traced it on this side, but I left it clear on the other side. Notice the mucous membrane isn't a straight shot, a straight line through. There are many folds within the mucous membrane of the vaginal canal. Why would these folds be present? What's the advantage of having these rugae-like structures? All right. The ability to be able to accommodate. You've got to pass a basketball, right? I know, be able to accommodate, and all the men puff up when I say that. No, I'm not talking about to accept the penis. I'm talking about to let the baby come out afterwards. However, is there another advantage to the folds in this mucous membrane?
Is it to keep the sperm there for longer?
It does increase the surface area so that it could help to keep the sperm in there. However, remember, the goal isn't necessarily to keep the sperm in the vaginal canal. It's to let them to go through. But you're getting along the right idea. It is a copulatory organ. The goal of copulation is to fertilize the egg. And to fertilize the egg, semen must be released. So by having these folds increasing the surface area, it increases the friction that is produced, right? It's ribbed for his pleasure. Because again, if you can help to stimulate that orgasm, feed that positive feedback mechanism, get the ejaculation to occur, then the semen can be released and the species can be propagated. So it serves both functions as a copulatory function, but also to accommodate the baby as it is coming out. Excellent. Oh, one last thing. Notice this illustration doesn't show it, and I'll clear it out. However, in females, at the time of birth, there is an enlargement of one of these folds in the mucous membrane that mostly, but does not completely close the vaginal canal. Right? It needs to be left partially open so that the menses can be expressed. But what do we call this enlarged fold in the mucous membrane near the distal end of the vaginal canal? Yeah, it's the hymen, absolutely. It is typically a well-vascularized and well innervated structure, making it very sensitive so that when insertion occurs, often from intercourse, or it can also occur from the insertion of a tampon. Tampons can be inserted without breaking the hymen, but if you're not careful, they can tear the hymen. And the hymen also can vary in its consistency. For some individuals, it is very thin and very flimsy. Something as simple as riding a horse or vigorously running or something like that can actually cause it to rupture. In others, it is much thicker and much more vascularized, and so much so that it is incredibly painful to try to tear it, and it actually has to be removed surgically before intercourse can take place. So it can vary in its size and its sturdiness, I guess would be the right word. Consistency, I think, was probably a better term, but there you go. Oh, great. One last thing. Let's go back to the picture. This is indeed a mucous membrane open to the outside world, but the epithelial tissue of the vagina doesn't actually contain mucus glands itself. Again, this is a copulatory organ whose job is, right, having that friction, having that copulation take place. And so we don't necessarily want openings in it that could get into the inside world. However, we do need to protect it. And so that protection primarily comes from mucus glands that are located in the head of the cervix. So it is a mucous membrane and it is lined with mucus, but the mucus that lines the vaginal canal is actually produced by the head of the cervix. It is mucus, catching dust and debris, providing that protection, acting as a lubricant, but it is also a very glycogen-rich mucus. Why might we want a large amount of glycogen in the vaginal canal? What do you know about glycogen?
It's sweet.
Okay, true, it's sweet, but it's probably not for taste. But what else do we use glycogen for?
Energy.
Energy. And in particular, as we know, the surfaces of our body, including the internal surfaces, are covered with resident bacteria. And when bacteria break down glycogen, do they use oxygen and completely break it down in a very efficient process of that aerobic respiration? No, they break it down using glycolysis. And as we know, when you use glycolysis, you produce pyruvic acid that gets converted into lactic acid. Remember, way back when we were talking about our body defenses, one of the body defenses we talked about was the acid mantle of the vaginal canal. By having that bacteria in there, that resident bacteria that's not harmful, by having that lactic acid in there, it is very, very acidic. that acidity does two things. The good things is that it helps to retard harmful bacterial, or let's say retards pathogen growth, keeping pathogens from being able to get in there and cause problems. And that is absolutely positively a good thing. However, as we also mentioned, that acidity slows the swimming of the spermatozoa. Now, one of the interesting things about this acid mantle is the vaginal canal doesn't actually start acidic. Earlier in adolescence and in the teenage, it starts more alkaline. Then, as the woman matures and ages, it becomes more and more acidic. You may not think of it in these terms, but females, when they're younger, when they're in their teenage and adolescent ages, they're much more likely to get things like urinary tract infections because of the lack of the acidity that is in that area. And as you know, teenagers basically just have to look at each other sometimes and they can get pregnant. Whereas, as we talked about, older people may have much more challenging issues trying to get pregnant as a result of that. So it is that lack of an acid mantle or decreased acid mantle that may actually stop fewer sperm, causing more sperm to get through, might be one of the reasons why there is a higher likelihood of pregnancy as an adolescent than as an adult. All right, the external os or orifice of the vagina is just one of the structures that form the external genitalia of the females. Thankfully, a more common term for these structures collectively is the vulva. As we kind of already mentioned, vagina gets its name from the term sheath, because again, it holds the sword. And as we've talked about, most anatomists are male, especially the ones that were naming these things. And even to this day, well, I say even to this day, but female reproductive research, sex research, all these things have huge taboos on them. And as we're seeing in the Supreme Court right now, as we speak, right, female reproductive rights and the rights of her body are things that are not apparently God-given rights. So one of the ways you see that is an older term they used for the external genitalia and the vulva is the term pudendum. Again, you're not going to be responsible for using this on the exam. It is an older, antiquated term, but it comes from the root of the same word that basically means shameful. So again, it tells you, gives you that appreciation of many of the challenges associated with females and the reproductive system. So yes, so thankfully that is not a term that is used really much anymore at all. And instead, external genitalia, or collectively the vulva, is a more common term. Let's start with some of the external features. Notice on the pubic symphysis, remember hopefully over Thanksgiving break, you found somebody, a loved one, some stranger at a bar, grandpa or whoever, where you were able to palpate their ductus deferens as it went over the pubic symphyses of their male. In females, there is a fat pad that actually sits on top of the pubic symphysis. And again, at the onset of puberty, when we're getting the growing of secondary hairs, when we're getting the increase in muscle and bones in males, you are also getting an increase in adipose in females. As we mentioned, that adipose is very important for the establishment of the ovarian cycle. This is seen in the hips. And one of the areas it's seen is over the top of the pubic symphysis, forming this round mound called the mons pubis, the round mound over the top of the pubic bone. This is where the pubic hair starts to grow in this area. So again, it is that fat area, and that swelling is an indication of sexual maturity. Notice also there are not one, but two specific folds in the skin that basically form the boundaries of the external genitalia. They are the labia majora. The labia majora is this outer region that we see here. This is a heavily keratinized stratified squamous epithelial tissue like the rest of the skin. Notice, like the rest of the skin, this is an area where our hair can grow, and secondary hairs do indeed grow on these outer skin folds. However, you can also see inside of that are these thinner, smaller, more delicate and hairless skin folds. And these two additional skin folds are what are known as the labia minora. And these labia minora basically bound the 4 orifices that are associated with this vulval region, with the external genitalia of the female. Notice this particular illustration only shows 2 of them. The 2 that we can see right here is the external os, or the urethral opening for the urethra, and the vaginal canal. And notice they've drawn what they think is looks like a fold of the mucous membrane, representing the hymen there in the vaginal orifice. So we can see a bit of that there as well. And so these are defined in here. Notice there are 2 more openings that are not seen here, but let's go ahead and erase those 2 things, and I'll tell you where they are. There are 2 additional glands that are located posterior and lateral to the vaginal orifice. And those 2 openings are openings to a couple of paired glands that we'll talk about in just a minute. So those 4 openings, the 2 openings to the glands, the urethra, and the vaginal orifice, are all contained within the labia minora. Notice anteriorly, the labia minora extends to form the prepuce, the foreskin over the top of the clitoris. Much like the male, there is an erectile tissue structure with an enlarged head, a glans. And again, in this case for the female, it is a clitoris. And like the foreskin, like the male has a foreskin, there is a foreskin around the clitoris as well. And again, the appropriate anatomical term is the prepuce. Unlike in males, where the prepuce is removed for cultural and cultural, religious, or medical reasons, unfortunately, in some regions of the world, the entire clitoris and prepuce can be removed in what is basically genital mutilation, but removed from females because, of course, they should not be allowed to enjoy sex, and any pleasure that they gain from it is pure evil. So those must be removed so that females don't cause males to give in to their sexual desires that can't allow them to be controlled. And again, as ridiculous as all of that sounds, there are plenty of societies and cultures where that is exactly what's happening. All right. I apologize for all of my disdain today with all of these topics, but obviously, like I said, what's going on with the Supreme Court, what's going on with a lot of these things is as a father of 2 daughters, is incredibly depressing. And for you, for the females in the class, you should be angry. All right. This space containing these 4 openings is what is known as the vestibule. Notice all of this is contained in a structure that we see. Good. I'm glad I'm not the only one. This diamond-shaped region that is known as the perineum. And this perineum basically, as I mentioned, contains orifices, and we'll talk about that in a second. The urethra, which we see here. The vaginal canal, which we see here. And here, now they show us these 2 additional openings that are the greater vestibular glands. Notice this perineum that we see here is something that a male has as well. And basically just think of it from, if we remove the pesky person, which I think I have done on the next slide. We'll get to all this in a second. Oh, I don't have the whole thing. All right, never mind. We'll come back. As you can see here, basically the four boundaries are the pubic symphysis, the coccyx, and the two ischial tuberosities. So from those four bony features, you have this diamond-shaped region that contains all of these structures in the female, and also the reproductive structures, as well as the anus in both males and females as well. All right. Let's talk about the glands first. We'll come back to the glands, the DS, glands. The clitoris, like the corpus spongiosum, is a long tube-like structure. It's actually really, it's an interesting combination. It's a combination of the corpus cavernosum and corpus spongiosum. Let's make this a little bit smaller. Too small. The corpus cavernosum, as we know in a male, basically is the larger structure. It does not have the urethra pass through it. But remember, the corpora cavernosa at the proximal end tapers to form crua, the plural of crus. And that's what we're going to see here with the clitoris as well. But it is also similar to the corpus spongiosum in that it is an elongated structure. And at the distal end, it enlarges into a glans. So here, if we remove that pretty picture of the person and just look at the tissue underneath, we can see this. So notice here, we can see there is the body of the clitoris, long tube-like structure. It has an enlarged head. But notice also it has the two tapered tails, a crus on both sides. So those two collective crua, and just like the corpus cavernosum, those crua help to anchor this structure to the pubic symphysis. Now notice the vast majority of these structures are internal. So when we look at the perineum in the female, when we look at the vulva, we see only the glands sticking out with that little bit of the prepuce wrapped around it. So these are a much more internal structure. Notice also, they're not the only erectile tissue that is found within the female reproductive organs. Here, we see these two enlarged bulb-like structures. These two enlarged bulb-like structures are indeed the bulbs of the vestibule, and they sit inside of the labia majora. We'll talk about arousal and orgasm in females, just like we did in males. But like a male, upon arousal, all of this erectile tissue becomes engorged with blood, making the clitoris typically stand out more, become more prominent, become more sensitive. But it also engorges the labia majora, with the advantage, again, the point of this is to be a copulatory organ. So by engorging the labia majora, it helps to form a protective seal around the penis during copulation, maintain the penis inside the vaginal canal during copulation, and helps to maintain the ejaculate in the vaginal canal after orgasm has occurred for the male. So again, all of these things help to maintain the copulation and then also to keep the semen in there. But what about the clitoris? The clitoris necessarily keeping the penis inside of the vagina? Probably not. So what would its function be? What is the function of the clitoris? I know on the surface these things are kind of awkward to talk about, but this is an anatomy and physiology class. This is where we're allowed to do this. Well, if the goal is to populate the species, do you want having sex to necessarily feel like you're having your audit on your taxes? Or would you prefer it to be a more enjoyable type of experience? These are not the hard questions.
Enjoyable.
Yeah, obviously, if you want to populate the species, you want it to be something that the female is going to want to participate in. So obviously, one of the advantages of having a heavily innervated clitoris is that it can be enjoyable for females. As we've also hinted at, if a female orgasms, that can produce a reverse peristalsis that can help to facilitate fertilization of the egg. But it is also believed that the enlargement and the extension of the clitoris may also help to stimulate the edge of the penis as well. Again, the same way that the folds lining the mucous membranes can help to stimulate the penis. Since the goal is to get that ejaculation from the male, anything that can help to facilitate that is a good thing. I want you to see, we're going to talk about it next. We did that. All right, we'll come back here. So as I mentioned, here in the vestibule, there are 4 orifices. 1, 2, 3, and 4. We know the function of the vaginal canal. We've just finished talking about that. We know the function of the urethra. We've just finished talking about that. However, if you notice, our artist cheated a little bit here. If you look really closely at this image, you see that there are 2 dots that they put there on the edge of the urethra. I'm going to remove my dots now so you can see the dots that the artist put there. Those are supposed to represent the opening to a paired gland structure known as the paraurethral glands, or what are also known as the Skene glands. Now, instead of this picture, I'm going to go back to our model. In our model, there are these 2 paired glands that are located within the wall of the levator ani muscle. That's what I need. And their ducts feed into the urethra. What does that sound an awful lot like? Didn't males have a pair of glands? Didn't males have a pair of glands, 2 glands that were located in the urogenital diaphragm with a duct that fed into the urethra?
Was the bulbourethral?
Yeah, it was the bulbourethral glands. Absolutely. So it sounds like that. What were the bulbourethral glands used for again?
Didn't they produce the clear mucosal, like alkaline substance?
Yeah, that pre-ejaculate substance, absolutely. They became active upon arousal to neutralize acidity, to aid in lubrication. And while these may look like the bulbourethral glands, the paraurethral glands aren't active upon arousal. Or again, what are known as the Skene glands. do not become active upon arousal. Instead, they actually become active upon orgasm. And they produce a secretion on orgasm. That secretion can, as you can see from the illustration, enter into and even out of the urethra. Oh, that didn't work because I'm writing in black over the top of black. There we go. And that secretion being released by the urethra may even give the female the sensation that she needs to void or urinate or is urinating during orgasm. However, is urine actually coming out of her urethra when she orgasms? No, it is this substance produced by the paraurethral gland. These substances have been studied, and it turns out they are very incredibly similar to the prostate secretions in males. I think Hannah was the one who said that prostate, this is actually the homologous structure to the male prostate. Again, how much of this secretion is produced can vary from person to person. Some people can produce a larger quantity of it. And like I said, can actually be expelled similar to a urine, but like a urine in that it can be expelled, but it is not urine that is released during that process. But there's one other way this paraurethral gland is similar to the prostate. Remember, as we mentioned, the prostate is heavily innervated. And for some males, the palpating of the prostate can be an incredibly enjoyable experience, leading to very intensive orgasms. Well, turns out the exact same thing is true for females as well. Notice the location of this gland is between the vaginal canal and the urethra. So once again, if you were to insert a finger into the vaginal canal and palpate the anterior wall of it, stimulation of that paraurethral gland for some individuals can produce a very intense orgasm, what people commonly refer to as the G-spot. So that elusive G-spot is really for some women just that paraurethral gland, and that innervation of it that leads to that increased sensitivity. So again, if you're not sure if you've got one or not, have you or someone you care deeply about insert a finger about 1 inch into the vaginal canal and palpate the anterior wall and see what happens. All right. Or do it during the next break. All right, questions on that? All right, so notice this gland doesn't actually feed to the outside of the body directly. It feeds into the urethra. So notice when we go back to this illustration, these two dots represent the openings to that paraurethral gland, but that's not actually where the openings are. They actually feed into the urethra. However, these two are openings within the vestibule, and those, as I mentioned, are the greater vestibular glands. Notice if we, again, remove all of the pesky skin of the person out of the way, we can see these big glands here feeding into this orifice. And these greater vestibular glands, they produce a thick alkaline mucus secretion, and they produce it at arousal. What does that sound an awful lot like? which
The bulb.
glands? Go ahead.
Urethral?
Yeah, the bulbourethral glands in males. This is the homologous structure in females. To the bulbourethral. Again, notice females basically have a homologous prostate. They have a homologous bulbourethral gland, a gland that becomes active upon orgasm, a gland that becomes active upon arousal. What they don't have is a gland like the seminal vesicle. But remember, the seminal vesicle is all about getting the sperm active and ready. Since female aren't producing sperm that way, they don't need a seminal vesicle. But these other two glands, they do have homologous structures. So the same way that, remember, the labia majora become the scrotum in the males, the greater vestibular glands become the bulbourethral glands in males, and the scheme gland or periurethral gland in females become the prostate in males. And really, that's probably the more accurate way to say that, because the default setting for our organism is female. It is the specialized genes that are in that Y chromosome that modify us from the default setting and to all those specializations that make a male. So when we talk about these homologous structures, remember they're one thing in males and one thing different in females, but obviously they come from the same cluster of cells. So the same cluster of cells in boys become the scrotum, in girls become the labia majora. The same bundle of cells that become the greater vestibular glands in females become the Cowper glands or the bulbourethral glands in males, and so on and so forth. So these are all examples of these homologous structures. So we did all of that. And how are we on time? We're a little over, but we'll do this. Not surprisingly, arousal and orgasm, we still have our point and shoot in females. However, there is obviously one big difference. Is orgasm required for fertilization in females?
No.
It's not required for fertilization, but it does help. As we talked about, it can produce those reverse peristaltic contractions that can help to draw the sperm up. So it can help, it can assist in the process, but it is not required, right? Obviously, male orgasm is required, but we've got the same kind of thing. It is going to be dealing with glands and smooth muscle. Arousal in females, just like in males, is a parasympathetic reflex. We get smooth muscle. We dilate the blood vessels to the erectile tissue. And the erectile tissue becomes engorged. Notice the clitoris and the bulbs in the labia majora do not have the same collagenous fibrous structures around them. So they enlarge, they engorge, but they don't become rigidly hard the same way that the penis does, because they don't have that tunica albuginea, that collagenous connective tissue that wraps around them. So again, that is our main, one of the main places where we get our smooth muscle. We affect smooth muscle by dilating those blood vessels. We affect glands. Which glands did we say became active upon arousal? Well, you've got two choices, greater vestibular or periurethral. Which one became active on arousal?
Greater vestibular.
There you go. And also those glands in the head of the cervix. Again, these are producing an alkaline rich, mucus rich, lubricating secretion to help to facilitate copulation. This will produce minor contractions of the vaginal canal and the uterus. Any other smooth muscle, and again, this is true for males as well, though it may not be as noticeable. Any other smooth muscle that contracts upon arousal? How about the smooth muscle in the nipples? Causing the nipples to become erect upon arousal. And again, this happens for males as too, it just might not be as noticeable, or at least nobody cares as much, but males' nipples can become sensitive and erect from the smooth muscle in that, as well as for females. And then, like in males, orgasm is a positive feedback, sympathetic reflex that again is going to involve smooth muscle and glands. In this case, we are going to get powerful contractions of the vaginal wall and uterus. And again, here they do this in a reverse peristalsis. To draw the sperm towards the uterine tube. And as we also mentioned, you can get an increase in glandular production, not just in the cervix and the greater vestibular gland, but you can also get activation of that periurethral gland as well. Like I said, in some cases, it can produce a larger quality of secretions that can be released. In others, it may just be enough to, again, by lining the surface of the urethra, may give the sensation of the needing to void, or the feel of voiding taking place. But again, it is not urine. It is a separate secretion, very similar to the secretions produced by the prostate in the male. And of course, what's the other big difference between males and females when it comes to orgasm?
The refractory period?
Yeah. Is there a refractory period for females?
No.
Yeah. Because they do not need to maintain the erection of the clitoris the same way the male needs to maintain the erection of the penis for multiple copulations or multiple orgasms to take place. The females don't have that, so many women can experience back-to-back orgasms if so motivated. All right. Questions on that? All right. That is everything I wanted to talk about for the male and female anatomy and function for now. As I mentioned, we do need to talk a little bit of development and inheritance. And once again, as I've talked about, learning biology in general, but especially anatomy and physiology, and especially genetics, is like learning a whole new language. So there is some vocabulary that I want to talk about and I want to get out of the way. And also, we've fallen behind on our histology. So we need to go back to the histology of the male, and then also go to look at some of the histology for the female as well. So we've got a lot of histology to look at. I have a little bit more of an introduction into what we want to talk about on Tuesday. So we need to get some of the vocabulary out of the way so we can hit the ground running that way. So we still have a fair amount to cover. So we'll take one more break. And when we come back from the break, where should we start? We'll start here. and then we'll go to this. We'll finish the rest of our introduction. We'll do our introduction first, and then we'll finish up with the histology so that we have time to do that. So let's go ahead and take one more break. It is 2:25, which makes it easy. That means we'll come back at 2:40. And I will see you guys, and we'll restart the recording at that time. And I will see you in 15 minutes. We're done with all the fun stuff of talking about reproduction, and now we just get to talk about the implications of it. Again, as I mentioned, I wanted to start first by focusing on some terminology. Again, gestation period is the period of time that is considered for the embryo to grow, and it is kind of a magic number because when gestation period is counted, it is counted from the last menses to birth, and it's about 40 weeks. However, as we know, ovulation doesn't occur until about day 14. The egg can be viable for up to 24 hours after that. And sperm, I don't know if we talked about this. Most sperm last 24 to 48 hours, but there are some longer lasting sperm that can last up to 3 days, 72 hours. So you've got a 3 or 4 day window there between when ovulation and copulation occur, where you can get pregnant. And again, once that egg is fertilized, it's instantly already 2 weeks old, because again, it's considered from the last menses to the point of birth. Those 40 weeks are divided into 3 main trimesters where different events occur. Prenatal period is the period, everything up prior to birth. And it is primarily divided up into 2 main biological stages. This is the embryological development of the first 9 weeks. And during that period of time, the developing ball of cells is considered an embryo. And then starting about 9 weeks until birth is considered fetal development, because that ball of cells is considered a fetus. Now, the main distinction between these 2 phases, obviously there are some big developmental stages, but really one of the primary determinants of the embryonic versus the fetal stage is basically where the hormones are coming from. As we talked about during the first 9 or so weeks, it is primarily the corpus luteum that is producing the hormones that are driving the process of the growth and development that are taking place. Whereas once it becomes a fetus, starting about week 9, the placenta has formed. And it is the placenta that is responsible for producing the hormones that are driving the developmental process. So this 9-week period isn't just arbitrary that, what's an embryo and what's a fetus. It's actually driven by where really the hormones that are coming from that are driving the process. And it's really the production of the placenta and then the hormones produced by the placenta that lead to that fetal stage of development. The neonatal period is basically the first month after parturition, birthing takes place. And of course, the individual who is primarily responsible for handling pregnancy, labor, and the neonatal period is an obstetrician. Most obstetricians, but not all, are also gynecological doctors as well. So the field of OB/GYN, you may hear a lot, is obstetrician and gynecology. Now, as I mentioned, we have a whole semester's worth of material and more that we could be covering. So what we are primarily going to focus on are the events that you are seeing right here. We've already pretty much started this process in talking about the ovarian cycle, which involves ovulation. the goal of getting that oocyte into the uterine tube, into the ampulla, where then it can be fertilized by the sperm. And then, as we mentioned, that fertilized egg, that zygote, is going to continue to divide, taking a mystical, magical journey into the uterus until finally it implants into the wall of the endometrium of the uterus. And so these are going to be the stages. These are going to be the things that we are going to talk about primarily on Tuesday. All right. So that is our game plan. That is our vocabulary. So we can go right into this on Tuesday when we meet. However, what we still need to spend a fair amount of time doing is finishing up the histology. We fell a little behind, so this is going to be a little backtracking. Because if you remember, the last time we looked at the tissues, we were still inside the male reproductive organs. And someone remind me what organ we're looking at it right here.
Testes.
Is this the testes or is this the testes? So is this one the testes or is this one the testes? Which one's this one?
Is the epididymis?
Yeah. Notice again, you absolutely did the right thing. The first thing you see this tube, you see all the nuclei. It's the first thought is 100% absolutely this must be a seminiferous tubule. However, how do we know that this isn't the testis and this isn't a seminiferous tubule?
The nuclei of the cells.
Right. Yeah, notice all the nuclei are near the base. They're not changing in size and shape the same way that they would during that. And there's practically no nuclei. There's one or two random cells in here, but there aren't very many random cells. And probably those random cells were probably damaged during the cutting process, so they probably don't even belong there. Is there anything else that is a giveaway?
The sperm shows in the slide.
Yeah. Absolutely. You may be tricked into thinking all these hair-like structures that are extending out are tails, but notice they can't be flagella because there are no nuclei associated with them. So these are those non-motile cilia that we talked about. Instead, as you mentioned, because of the dehydration, because of the way this tissue is processed, all of the sperm are going to condense together in the center of our epididymis. So it is a dead giveaway. Notice even here, we notice it as well. Again, that's clumping together of the cells, the lack of cells in the innermost layers of it, and the lack of the change in the shape. Again, notice it's very distinct. We have the stem cells and then the super big primary, and then the secondary gets smaller, and the spermatocytes get even smaller, or the early spermatids get even smaller, and then the late spermatids are sperm shaped. We see this very clear, very stereotypical change in the nuclei in the seminiferous tubule that you just don't see in the epididymis. So again, yeah, you absolutely did the right thing. The first thing is to think, boom, that's got to be the testis, that's got to be a seminiferous tubule. But when you look closely, you see that it is not. All right. Again, sperm, pretty simple and straightforward. The only thing I really want to remind you is notice on many of these, on the apex of it, on the head, you can actually see that acrosomial cap we were talking about. Notice slightly lighter in color on the top of most of the heads of these sperm, because that is where that acrosomial cap is. You can actually really see that big, clear digestive enzyme capsule sitting on the top of the sperm. And again, notice here, it's much lighter in color on those as well. Not a lot of other anatomy you need to really worry about for those. Other than knowing the regions, obviously we've got the head, we've got the mid piece, and we've got the tail. So again, pretty simple anatomy for that. What we didn't look at that I wanted to look at, because we talked about it, is the penis. Now, this penis doesn't happen to be the penis of a human. This is, I believe, a rat penis. So one of the things you will notice is, again, we see those 3 layers of spongy tissue that we would expect to see. The 2 lateral and superior corpora cavernosum. And around the urethra, we have the corpus spongiosum. And notice, and again, that's why I like this picture and I use this picture, notice how thick the tunica albuginea is. We can see this big, huge, massive collagenous fibrous structure wrapping around this, helping to, again, so that as these engorge, they push against that boundary, they form that congestion, and they make this a rigid copulatory organ. This one is actually a human penis. Notice as we look at the human penis, notice the corpus spongiosum is much larger around the urethra, very clear there. Notice we can still see the tunica albuginea, but it's not quite as pronounced as it was in the rat one. And we can see a little bit of it along the midline here. But what I also really like is we can also see, and I'll change colors here. Notice we can actually see one of those deep arteries that is feeding the blood. So I'm going to go ahead and erase that so we can see it now that you know what I'm here. I'll just point an arrow at it that way. I don't have to obscure it. We can see that deep artery that is actually feeding the blood into this spongy erectile tissue to help to, again, cause it to engorge and become that copulatory organ. All right. Questions on that? No, one more. Here, notice this one. Again, really, really nice deep arteries that can be seen very, very clearly in the tissue. Clearly see the distinctive difference between the corpus albicans, pardon me, not the corpus albicans, the tunica albuginea and the erectile tissue that is in there. And notice this one looks like it may be near the tip of the urethra, because this looks, again, I wouldn't ask this on the exam, but this looks more like a non-keratinized stratified squamous epithelial tissue than it does a transitional epithelial tissue. Remember, for the majority of the urethra, it's going to be a transitional epithelial tissue, but near the tip, near the opening, it's going to be a non-keratinized. And again, at this magnification, I wouldn't ask you to be able to distinguish that, but I've looked at enough tissues that that looks clear to me. And one more of the urethra. All right. What I want to do now is switch gears to the female. Well, guys, let's go back. Any questions on any of this before we move from the male histology? I think that is everything you're responsible for on your histology list for males before we move to the female. So any questions on any of that?
Yeah, this might be like really obvious, but what's the difference between non-motile cilia and normal cilia?
Normal cilia, remember, move in a very distinct wave-like fashion to move substances across their surface, whereas these basically don't move. They're just there for surface area. They don't produce a wave-like motion. They're not capable of moving anything along their surfaces. And that's it. Just one move and one doesn't.
Professor, on this slide, since that's the end of the urethra or like around there, would that be like the mucus glands around? Yes.
So you are correct. Within the lamina propria of here of the urethra, you can see some of the mucus glands, absolutely. Whereas you can also see the spaces in the spongy erectile tissue that would wrap around it as well. Absolutely.
Thank you.
Yep. All right. Any more questions on the boy parts? All right, let's talk about the girl parts. This is an ovary from a cat. Now, obviously, cats give birth to litters and humans do not give birth to litters. But what they've actually done to this one prior to harvesting this ovary is they have injected it with follicle stimulating hormone. Notice when they inject this animal with follicle stimulating hormone, there's no longer a competition for follicle stimulating hormone. So as you can see, many of the follicles are maturing and developing. And that is why they do that. They do that to be able to see if this is maturing, if this is developing, and if this is maturing and developing, then that is an indication, then that allows us to see all of these follicles, which is the point of why they're doing that. Notice we see, based on this, a massive number of tertiary or mature follicles. Now notice with these mature follicles, I'm not seeing eggs in any of these 4 that I'm drawing circles around. Why not? Shouldn't this have an egg inside of it? Does this mean this follicle didn't have an egg?
Is it starting to become the corpus luteum?
That's not a bad guess. And in fact, if you look closely, indeed, we do see one example of a popped balloon where we can clearly see that this is one that has undergone ovulation and is collapsing, and the fluid inside is going and it's collapsing down. So this is this one here. And now that I've drawn around it, I'll erase it. That one here, this is the beginning of the formation of a corpus luteum. So is it possible that this is caught right as it popped? Sure, maybe, although you might expect it to collapse in on itself. But remember, you have this big, huge follicle that has a tiny little egg in it, and it's a 3-dimensional structure. If I cut through an apple, if I close my eyes and cut through with an apple, am I always going to hit a core? Is it possible to take a slice of an apple where you didn't see the core?
Yes.
Yeah, absolutely. So is it possible to take a slice, a 2-dimensional slice through a 3-dimensional structure where you don't see the eggs inside of these follicles?
Yes.
Yeah, absolutely. So yeah, we don't see the egg, but obviously we see the huge, massive antrum. And so clearly these are mature follicles. And yeah, we got lucky. That's the advantage of having so many of them. Some of them absolutely are going to have eggs inside of them that we can see. Here's a really, really nice one that we can see all of the nice anatomy. And so that's why they do that with a lot of these. Here we have a nice secondary follicle that we can see here. This is a good secondary follicle. We see plenty of primordial follicles. This is probably a primary that we can see there. So all of these at different stages of development, that's why they do this, so we can see lots of examples. Here's another example of a cat ovary where they've done the same thing. And again, we see many, many mature follicles because they've injected it with follicle stimulating hormone. We talked about, I drew them, we discussed them, but let's actually look at cells in the different stages of the ovarian cycle. What would this entire cluster be known as?
Primordial.
True, each individual structure inside of it is a primordial follicle. So indeed, this is a primordial follicle, and this is a primordial follicle, and this is a primordial follicle, and on and on and on. There's probably a dozen in here. But these dozen together, what do we call these dozen primordial follicles together? You guys keep getting the hard questions right and getting the easy questions tripped up on. What do you call a bunch of eggs clustered together?
A nest?
Nest. These are egg nests. Absolutely, this is a nest. This is a nest of primordial follicles. Notice we can very easily and clearly see there is the cell at the center. And how would you identify the cell at the center? Be specific. I guarantee you this question is on the exam. Gladly. Identify the cell at the tip of the pointer.
Is that oocyte frozen and mitose?
Okay, you get partial credit for that on the exam, but give it all to me. Again, I will have the words be specific there on the test to help you to know to be as precise as you can be. So give it all to me. I heard oocyte. I heard frozen. What can we add to this?
Is it prophase 1?
Okay, excellent. So it's frozen in prophase 1. Excellent. But are we going to get away with just calling it an oocyte? There we go. Amy's got it. A primary oocyte frozen in prophase 1. I want all of that. If I ask you for this cell, I want all of this. It is a primary oocyte frozen in prophase 1. And we can see nicely that there are this single layer of flat cells around all of them. How would we identify these cells that I've drawn in blue?
Simple squamous epithelial tissue?
Okay, they are simple squamous cells. And I'd accept that, but didn't we have a name for these simple squamous cells?
Granulosa.
They're not granulosa yet. When they're simple squamous, they're not really producing estrogens yet. And at that point, they're still just called follicular cells. Right? That one layer of simple squamous cells are follicular cells. And these two things together, the primary oocyte frozen in prophase 1 and the one layer of follicular cells collectively, what is that entire structure? And I know we've said it already, but give it to me again. Excellent. A primordial follicle. So now let's do it this way. I have a primary oocyte frozen in prophase 1, single layer of follicular cells forming our primordial follicle bound together in an nest with other primordial follicles. Now, notice we see a ton of primordial follicles in here, and then something interesting is happening right here. What's going on right here?
Follicular cells enlarge, become cuboidal.
Excellent. So notice I have one layer of cuboidal cells. Excellent. And what do we call these cells once they become cuboidal cells? Come on, someone said it earlier. What are the cuboidal cells? There you go, granulosa cells. So I have one layer of granulosa cells. Oops. That are cuboidal. They are wrapped around an egg. Identify this cell, be specific. And this is why I'm glad we're doing this. Better we struggle with this now than on the actual exam. Identify the cell at the tip of the pointer. Again, a question I guarantee is on your lab exam. Be specific. Is it a secondary oocyte at this point?
It's still a primary oocyte.
Yeah. And it is still frozen in prophase 1. So I have a primary oocyte frozen in prophase 1, and now a single layer of granulosa cells. So what is this entire structure equal?
Primary follicle.
There you go. Excellent. Again, see some more examples. More examples of primordial follicles. Cells with flat cells around them. Notice here, we have an example, not a good example, but an example of a primary follicle. And then we have this thing over here. Let's do it again. Give myself enough room. Identify the cell. Be specific.
Is it still granulosum? So,
Well, okay, I'm not pointing at that, but I'll give you that one. If instead I'd put my arrow here, then you're absolutely correct. Now we would have 2 or more layers of granulosa cells. Absolutely. So that'll give you, but that wasn't where my arrow was pointing. My arrow is pointing to this big thing in the middle. What is this big thing in the middle? Identify this cell, be specific.
Is it a second? Yeah.
Go
Go
ahead.
ahead.
it's secondary oocyte.
Not yet. So what does that make it? There we go. Still a primary oocyte frozen in prophase 1.
So is it antrum that you're pointing at, or it's the whole entire thing is a cell?
I'll move the arrow. This whole thing is a cell, and we can actually see the nucleus. So this whole big clear thing is a single cell.
Oh, I thought the nucleus was the cell, and
No, no. This is just the nucleus and a nucleolus. Notice it shows some protein synthesis might be going on. Oops. Oh no, didn't want to do that. Excellent. No, this whole thing is the cell. And it is much larger, but it is still a primary oocyte frozen in prophase 1. Ah, dang it. Oh, there we go. So now I have a primary oocyte frozen in prophase 1, but it is surrounded by 2 more layers of granulosa cells. So what do I call this entire structure?
Secondary follicle.
There you go. So notice, yes, the cell gets bigger between a primordial and a primary and a secondary. It's growing, it's getting larger, but it has not completed meiosis yet. So notice in the primordial, primary, and secondary follicle, the egg doesn't change. It's still a primary oocyte frozen in prophase 1. So really, the only difference between these three follicles are the cells that surround them. If it's a single layer of flat cells, it's a primordial follicle. If it's a single layer of cuboidal cells, it's a primary follicle. If it's two or more layers of a granulosa cell, it's a secondary follicle.
Professor.
Yep.
Can you go over how you would ask the question between us giving the answer as a secondary follicle and primary oocyte frozen in prophase 1? Like what you would point to?
Yep. So, identify the entire structure.
Secondary follicle.
Identify the cell, be specific.
Primary oocyte frozen in prophase 1.
Identify the cell.
Granulosa.
There you go.
Thank you.
Yep. All righty. I've taken up a lot of the screen. I don't know if I'll be able to still get away with this, but let's see what happens when we flip the screen. Because I kind of like what we've been doing up here, so I kind of want to keep it, but we'll get rid of it if we need to. Ah, excellent. All right. Notice again. Identify the structure. How is this different from the question I just asked?
Would it still be a secondary follicle?
Absolutely, it would still be a secondary follicle. Identify the cell.
Granulosa.
Excellent. Identify the cell.
Primary oocyte frozen in prophase 1.
Excellent. And one more. Identify the cell.
Fecal cell.
Excellent. And not only do we see that nicely here, but let's do one more thing. Identify the layer. Notice there's this clear layer between the granulosa cells and the egg. What is this clear layer?
Oh man, is it zona pellucidum?
That is exactly correct. Absolutely. All right, that glycogen-rich layer surrounding it. Awesome. Excellent.
So Professor, what would the corona radiata be?
Remember, to get a corona radiata, there's another great example of the zona pellucidum, another secondary oocyte. So remember, we need to have a mature follicle. Notice this is still a secondary follicle. We are starting to see, I don't like the green. We are starting to see some separation, some space forming. But look at the size of the space versus the size of the egg. The egg is still bigger. So this is not a full formed antrum yet. To get, to get to still secondary, we're starting to see those spaces that have formed. Ooh, really nice primary that you can see real nicely right there too. Secondary, all the examples, boom. Here, 5, 2, that. Is that going to cover it up? It did. Shoot. All right. We lost our primordial. It's still there. There's just no white. Here, let's right there. Yeah. I don't know how long ago I erased it, but oh well. That's the easy one, so we'll be okay. Notice here, now when we look at the size of the space versus the size of the cell, this is clearly a big, huge fluid-filled antrum. This is our tertiary follicle. And again, just like we talked about before, it is going to be made up of an egg in some stage and the support cells. So let's do the same thing we did last time. On the exam, if I had a nice big fat arrow pointing right here and asked you to identify that cell, be specific, what would you say?
Secondary oocyte frozen in metaphase 2.
Yep. Oops. Why is that not right? Excellent. Only when it reaches the mature state does it complete the first meiosis and start the second one. And again, notice it's got far, far more, but it's still 2 or more layers of cuboidal cells. And of course, these cuboidal cells we call granulosa cells. So we still have, but now we have a huge antrum. And as you mentioned, now some of these cells, the granulosa cells, are forming the boundaries of the follicle. We can't see all of it because it's under here, but here we see a full antrum-filled follicle without an egg for a mature follicle that we can see there. Some are forming the outer structure, but notice some of them are in direct contact surrounding the egg. Now, again, is there any difference between this granulosa cell here and this granulosa cell here? If I were to rip both of them out and look at them independently on a microscope, would they be the same or would they be different?
Same.
Completely the same. The only difference is their location. So here, the granulosa cells that stay around the oocyte, those are the ones that form the corona radiata. So notice you can't have a corona radiata without having an antrum. You can guess at it. Notice if we go back a couple of pictures, right? We can guess that these cells here are going to be the ones that are going to become the corona radiata, where clearly these have already separated from them and won't, but we still wouldn't call them a corona radiata here at this stage because we haven't got that full antrum yet, right? Even here, we can kind of get a hint at, all right, these are the ones that are probably going to stay in contact, and those might go away because we can see the space that is starting to open. But you don't technically have a corona radiata until you have a mature. There we go, beautiful. A mature Graafian follicle. Or tertiary. Again, all three of those terms interchangeable. Tertiary, mature, or remember, because good old Bob Graafian, probably Graaff, was the first one who discovered it, so it's a Graafian follicle after him. All three of those are acceptable terms. And it's made up of our secondary oocyte frozen in metaphase 2. And its follicle antrum. Again, that zona pellucida is so distinct. These granulosa cells are so compact. It's so easy to see that layer where they're not present. The fecal layer is a little trickier to see, but again, we can clearly see that there are cells on the outside of the granulosa cells that are wrapping around it. So these layers are super easy to see. Those parts are super easy to see the more mature the follicle gets. And here's another nice mature follicle. Then, as we mentioned before, and I'm going to go ahead and clear this. So any questions on the follicles? Do our 4 types of follicles, primordial, primary, secondary, and the mature Graafian follicles make sense?
Yes.
Okay, excellent. So then I'm going to go ahead and get rid of all of that. So, because it was getting a little crowded. As we mentioned, right after ovulation occurs, that follicle really does deflate. This is filled with fluid. And when that balloon pops, our balloon deflates. And so it starts by looking at this deflated balloon. But remember, these remaining follicular cells are being transformed by the corpus luteum. I mean, by the luteinizing hormone into the corpus luteum until it becomes this big, huge, massive glandular structure. Notice it went from a popped balloon to this huge, huge globular component. Big, typically very dense, because it is this massive, active glandular structure. Again, here's another one. This whole thing is this big, huge, look at all those cells in there. All of these darkly stained cells with their nuclei are actively producing massive amounts of hormones. Look, here we have a mature follicle. Look at the size of our mature follicle compared to this gland. It's this huge, massive glandular structure, releasing all those estrogens and those progestins. Until it shrivels up into something like this. I appreciate that this isn't the best resolution for an image, but what I liked about this is because it's a little bit granular, you can clearly see these very whitish areas. You can clearly see the areas that are filled with cells, filled with follicles. Mature follicles, secondary follicles, primordial follicles, all these follicles. And then there are these areas that really just look like this whitish scar tissue. And this whitish scar tissue is that corpus albicans, is that shriveled up corpus luteum that is no longer functional. Makes it really easy to identify, really easy because of that. Just again, it's basically clear. I mean, it's not clear as in like, you know, the antrum is clear, but there you don't see a lot of nuclei, you don't see a lot of cytoplasm. It's just this shriveled up whitish scar tissue. Very, very different from the rest of the uterus. And I believe, nope, lied. Uterine tube or fallopian tube, or also known as the oviduct. Remember, one of the things that I mentioned about it is that, and let's get up, I think I have a closer view. I guess this is the closest view I have. What is unique of the uterine tube is we have a simple columnar epithelial tissue that contains cilia. None of these are a great illustration of this. None of these, and again, these are things that I just threw together quickly because I wanted to share with you, and this is the best that I could find. but their job is to propel the egg. Now, if you think about it, the egg's not capable of propelling itself. So if you had this huge oviduct and you had an egg floating around in the center, if it wasn't in contact with the walls, if it wasn't in contact with the epithelial tissue, would it be able to move? These are the easy questions.
Oh,
No, absolutely not. So if you notice, one of the most distinct characteristics about the uterine tube is these big, huge, massive extensions of the uterine tube wall. Now, obviously, it looks like some big gaps here, but this is just the way it's been sliced and put on the slide. Notice, no matter where the egg was hanging out inside of here, whether the egg's here, or whether it's here, or whether it's here, or whether it's over here, it's going to be in contact with that wall. By having these huge folds, again, normally we think of rugae as things that allow for expansion. We don't need this to expand. But what we do need is to make sure that that egg is always in contact with the wall, so those cilia can do their job of moving the egg to the uterus. And so it makes the uterine tube an incredibly obvious tissue. Yes, it has, when we look at it at a high magnification, it is going to have a single layer of very uniform cells with columns and nuclei that all line up, and super obvious, super long cilia coming out of the top. Again, it's going to be a very unique and easy tissue to see that way. Well, you know what? I wonder, we were just on the site for, I haven't bothered to look, so I don't know if this will be any good or not. Whoops, where did it go? Come back. All right, you guys seeing the model? Yes, no? Are you seeing the website I'm on?
We saw it and then it went away. Okay.
Okay. Yeah, okay, so there is reproductive female histology on here. I haven't bothered looking at it, so I don't know if it's any good. Here we see, obviously, we're in the ovary. It's a pretty good mature follicle, another nice mature follicle. There we go. So there's our uterine tube, but I was hoping they would have, you can tell that it's lined up and uniform. I was hoping for something better. Yeah, so again, it's a ciliated, so they don't have a good picture of it either. The Yale site is a great histology site. They might have some good pictures there. But again, it's a very distinct tissue, so it's pretty easy to identify. But I think that now is everything that you're responsible for histologically for both the female and the male. All right, any other questions? All right, let me remind you, this is your big weekend to study. All right, next week is your exam. Your exam is 1 week from today. We will go over some new information on Tuesday. Again, it's going to be development. It is going to be, it will not be heavily emphasized on the exam because, again, obviously we're getting it 2 days before the exam, but there are some fun things we want to talk about that and mention. And again, obviously I clearly cannot do a whole semester's worth of development in just a couple-hour lecture, but we do want to touch on it a little bit. It's a nice, bow to put on the top of this reproductive system section. And then that's everything you'll ever need to know about anatomy and physiology. You'll take the test on Thursday, and then you have a nice 5-day weekend to prepare for your final exam. Tuesday's lecture will not take the whole class time. So we will have plenty of time at the end of that to be able to do a review. So I know this is an extensive review. I will cover anything that you guys want to cover, both for this coming lab and lecture exam for the reproductive system, but also for the final exam as well. So come with questions. Remember, if you don't ask questions, I've assumed you've mastered the material and I make the tests harder. So make sure you've got all weekend to come up with plenty of questions so I can help you to be successful next week on the exam. All right. Questions on any of that? All right. Have a good weekend. Be safe, be smart, and I will see you guys on Tuesday. Bye.